This is video 3 of our nucleic acid and protein synthesis chapter. We left off talking about RNA and we discussed transcription and how to complete it. Now we're going to talk a little bit more about RNA. There are 3 types of RNA, according to your vocab: messenger RNA, transfer RNA, and ribosomal RNA. All 3 are made by RNA transcription in the nucleus from DNA. And all 3 types of RNA are needed to make protein. RNA is the middleman between DNA and protein, and it is needed in order to make protein happen. So at the bottom here, we see our central idea for the chapter. DNA makes RNA, and RNA makes protein. Protein is what you see when you look at yourself in the mirror, when you look at other people. Proteins control all of your traits. So messenger RNA, which we call mRNA, this is the chain of hundreds of nucleotides in multiples of 3. Each group of 3 nucleotides is called a codon, and these codons determine the number, type, and sequence of amino acids that make up a protein. Amino acids are the monomers of proteins. So you'll see every 3 bases, every 3 nucleotides here is a codon. So AUG is a codon, GCC is a codon, and UAG is a codon. The first codon is always called the start codon, or what we call the initiation codon, and it is always going to be AUG. Always. Every single time. AUG. So as you're doing your transcription problems that we're going to be doing on, what you did on worksheet 3 and what we'll be doing on worksheets 4, 5, and 6, if your RNA sequence does not start with AUG, you're wrong. Just how it goes. Then you have your body codons, which are all the codons that fall after your start codon. But then the last codon, you're going to end with a stop codon, or what we call a nonsense codon. And that would be typically your last one. And it actually does not code for an amino acid. It just tells this process to stop. So when the ribosome is reading this mRNA sequence, when it reads AUG, it tells the ribosome, hey, we need to start building this protein. And it will continue to read that RNA sequence, that mRNA sequence, until it gets to a stop codon. Once we reach the stop codon, the ribosome says, all right, this protein is done. So once that mRNA sequence has been made in the nucleus during transcription, that mRNA can then travel out of the nucleus and to that ribosome, where it's going to be able to make that protein. Transfer RNA, or what we call tRNA, looks really weird. It's what you see to the right here. And it is a single chain of about 90 nucleotides, and it loops and bonds in a unique shape, which is what you see pictured here to the left. With the help of ATP and enzymes, the job of tRNA is to bring an amino acid, which you see up here as AA. It's going to take an amino acid from the cytoplasm and bring it to the ribosome to start building an amino acid chain, which is going to be your protein. The bottom of your tRNA, you'll see some more bases here. And this group of bases, there are 3, just like a codon. And we actually call this the anticodon. So in English, you've learned anti would be opposite of. So your anticodon is the opposite of your mRNA codon. And the purpose of an anticodon is so that the tRNA could land on the mRNA and drop off that amino acid, which you'll see in a couple minutes. The last type of RNA is ribosomal RNA, which we call rRNA. And essentially what this does is it makes up the ribosome. So you have a top part here called the 60S subunit, and you have a bottom part here called the 40S subunit. You don't need to know that. All you really need to know about ribosomal RNA is it is what makes up the ribosome in order for this process to happen. So we're going to look at this process of protein synthesis, which is also known as translation. So we transcribed in an RNA molecule. So in this case, we're going to talk about an mRNA molecule, okay, what you see in red. So this has been transcribed in the nucleus, and it is able to travel to the ribosome, which you see here in the background, all right? So it's going to run across the surface of this ribosome. And we're going to read this mRNA sequence, this message that has been brought to the ribosome. So we're going to call this process translation because we're going to read it, and then we're going to take that mRNA sequence and we're going to bring amino acids to that ribosome and make a protein. So we're reading and we're translating what it's telling us. So this process is called protein synthesis, and it's also called translation. So here we see AUG. So that's a codon, and it's going to code for a specific amino acid, and that amino acid is called methionine. So what's going to happen is a signal is sent out to the cell, and tRNAs are going to bring methionine over to this ribosome. And you'll see here's an anticodon. A pairs with U pairs with A, G pairs with C. It's kind of like landing gear. This anticodon is going to come to the ribosome and kind of land on top of this mRNA. And the purpose of that is so that methionine can be dropped off, kind of like cargo. Then the next codon is read. GCA codes for an amino acid called alanine. So the tRNAs go out into the cytoplasm and they bring back an alanine. Our anticodon here helps us land. And you'll see here that a bond forms between methionine and alanine. We now have two amino acids that are stuck together, which is called a dipeptide, which you learned about in a past chapter. And we're starting to build this protein. So after those are read, then we're going to shift this mRNA molecule into the ribosome further, and the next codon could be read. Okay. I'm going back here. This is dehydration synthesis, guys. We are building a molecule. So this tRNA pops off, and it's going to go back into the cytoplasm. Methionine is left behind. Alanine is here, and we're going to move this mRNA sequence back through the ribosome. So now we could read AAC, and that's going to code for an amino acid called asparagine. Our anticodon, A pairs with U, A pairs with U, C pairs with G. We use that as landing gear to drop off the amino acid. Go back. And then UAA is the last one that's read. That's actually a stop codon. So that's what tells us this process is done. This protein will only be 3 amino acids long, and that's all right. But this process is now over. Translation has been completed. So you might have been wondering, how do we know what amino acid is needed? How do we know? We know there are 20 amino acids in nature. We learned that a while ago. But how do we know it was methionine? How do we know what's going on here? Well, you have a tool that I've given you, and it looks like what you are seeing on your screen now. And this is the amino acid wheel. And this is how you're going to use it. So our first codon, I told you, is always going to be AUG. So what you do is you're going to start in the center with A, and then you move outwards. So U was our second letter. So we have A, we move to U, and then our last letter was G. So we move to G. And then that's going to tell me that the amino acid I need is methionine. Awesome. Let's try another one. How about U, U, U? U, U, U. That's phenylalanine. All right, let's try another one. How about G, C, A? G, C, A. That's alanine. How about UGU? UGU. That's cysteine. Cysteine. I think that's how you pronounce it. And then one more, AAC. AAC, that's asparagine. This is the tool that you're going to be using for worksheet 4, 5, and 6. And then I'll also give you one on the test that you're going to be using as well. You will also notice that there are certain codons like UGA that code for stop. So stop is not the name of an amino acid, but that would be a codon that would tell us to stop translation. You'll also see there are 2 other stop codons, UAA and UAG. So there are only 3 stop codons that exist. There are also many codons that code for one type of amino acid. For example, CCC gives you proline, CCU gives you proline, CCA gives you proline, CCG gives you proline. Okay, so those are all the ones here that give you proline. And I'm actually going to stop the video here because we're going to get into worksheet 4 next, and with the time I have to explain that, I will surpass my 15-minute time limit. So tune in to the next video for an explanation of how to do worksheet 4.